Hello everyone! Today our group Mia, Ming and Evgenia will be presenting our bioinformatics project on Yersinia pestis, the bacterium responsible for the bubonic plague. Our focus is on how different Yersinia pestis strains compare genetically and specifically how variations in the PLA gene may influence the bacterium's virulence. The bubonic plague is known as one of the deadliest pandemics in the human history. It likely began in China in the 1300s and spread rapidly along major trade routes. It is estimated that about 200 million people died of the plague. The organism behind these outbreaks is Yersinia pestis, a bacterium capable of causing three forms of plague: bubonic, septicemic, and pneumonic. Transmission typically occurs through flea bites, contact with infected tissue, or inhaling infectious droplets. Once inside the body, the bacterium can spread, multiply, and overwhelm the immune system. In our project, we wanted to understand how different strains of Yersinia pestis differ in virulence and how that might relate to the PLA gene. We analyzed 4 strains: 91001, which is not pathogenic, C2614, C2944, and CO92, a wild type and highly virulent strain. We hypothesized that the strains 91001, C2614, C2944, and CO92 of Yersinia pestis influence the performance of the protein PLA protease, affecting the virulence of the bacterium and its impact on the human body. We extracted and compared their PLA gene sequences and also compared them with similar genes from other bacteria like E. coli and Salmonella. By creating a phylogenetic tree, we are able to visualize how closely these genes are related. This helped us identify the role of PLA protease and why it is considered a major factor in the Yersinia pestis produces a protein called PLA protease, which is found on the outer membrane of the bacterium. Its job is to manipulate the human host's blood clotting system. PLA protease does a few things. It activates plasminogen, turning it into plasmin, which breaks down fibrin, the protein involved in forming blood clots. Without fibrin, the body's barriers weaken, making it easier to the bacteria to spread into the bloodstream and lungs. PLA can also break down or alter host proteins involved in inflammation and immune defense, contributing to tissue damage. And importantly, PLA becomes much more active at human body temperature, which increases virulence and allows the bacteria to multiply quickly once inside a person. Together, these changes help explain why bubonic plague progresses so rapidly. I will now have Mia discuss our project objectives and the research process.
All right, thank you, Evgenia. So I'll be discussing our project objectives. Our goal was to examine the PLA gene in different Yersinia pestis strains and compare these sequences with related protein sequences from bacteria such as E. coli and Salmonella. To analyze the similarities and differences between these proteins, we used a phylogenetic tree. Our goal was to understand how changes in the PLA gene might affect virulence and the bacteria's ability to spread. We also wanted to explore the evolutionary relationships of the PLA protease with other Omptin family proteins like the ones in E. coli and Salmonella. So there were 3 problems we really wanted to kind of identify and touch upon. So the first one we're going to discuss is that Yersinia pestis is deadly if not treated with antibiotics quickly, but we don't fully understand how small genetic differences can affect its ability to cause disease. Now, as discussed before with Yersinia, the PLA gene is a key virulence factor to Yersinia pestis, but it's unclear whether changes in this gene and in its different strains may affect PLA protease activity or infection severity. And number 3, understanding these differences is important for predicting how dangerous different strains might be and for developing better treatments or preventative strategies. So now I'll be discussing our research process. So to start off with, what we did is we investigated virulence factors of Yersinia pestis. We found the PLA gene and then used the National Center for Biotechnology, aka the NCBI, to obtain protein sequences from different strains of Yersinia pestis. We then used the basic local alignment search tool, also known as BLAST, to discover other bacteria with similar sequences to Yersinia's PLA gene. That's where we discovered E. coli and Salmonella. So once we used those websites and we were able to obtain those protein sequences, we made sure to save all their FASTA files for analysis. We then built a phylogenetic tree. We used the website phylogeny.fr to align the sequences and create the tree. That's where those saved FASTA sequences come in, as it is easy as pie to just copy, paste, and create a tree. So once we created our tree, we then needed to compare our sequences and looked for small changes or mutations in the PLA proteins, basically meaning we were just looking at the branches to see how closely related all of our bacteria were. Then we needed to analyze the results. We discussed how differences might affect PLA activity, virulence, and evolutionary relationships. I will now hand it off to Ming, who will discuss our results and our conclusion.
So after running our sequences alignment and the phylogenetic analysis, we found that the PLA gene is almost identical in all 4 Yersinia pestis strains we looked at: 91001, C092, C2614, and C2944. The amino acids sequences match extremely so close, and none of the tiny differences we observed fall into regions that are known to change how the PLA protease functions. This suggests that these strains mostly produce the same PLA protease, which means they probably use similar mechanisms to spread infections in a host. When we visualize the sequences on the phylogenetic tree, all our 4 Yersinia strains clustered tightly together. In contrast, the related genes from E. coli and Salmonella branched off much farther away. This tells us that while these bacteria share a distant ancestors, they are not closely related to Y. pestis in terms of the PLA gene. Overall, one of the biggest takeaways is that the PLA gene is highly conserved in the Yersinia pestis because PLA plays such a key role in breaking down host tissues and helping the bacteria disseminate. It makes sense that the genes will stay almost unchanged across the strains. Its conservation highlights just how important it is for Y. pestis survival and virulences. However, even though our findings show strong genetic similarity, our hypothesis ended up being inconclusive. We could confirm evolutionary relatedness and conservation, but we couldn't determine whether the small differences we did observe actually influence virulence. To answer the question, we would need additional laboratory experiments. For example, biochemical assays to measure enzyme activity, structure modeling, or infection studies to compare how each strain behaves in real biological conditions. So in summary, firstly, the PLA genes is almost identical cross of 4 pestis strains, and Yersinia sequences cluster closely together while E. coli and Salmonella are more distant. And PLA appears to be a highly conserved and functionally important virulence factor. And finally, the further experimental work is needed to confirm whether genetic differences influence how dangerous each strain actually is. Thank you so much for listening. This is our references.